Hello viewers, welcome to my channel. Today we will discuss about Japanese encephalitis. Japanese encephalitis is one of the main cause of viral encephalitis in many countries of Asia. It is estimated that almost 68,000 clinical cases occur every year. Japanese encephalitis is vector-borne disease as well as zoonotic disease. A vector-borne disease is one that is transmitted by arthropod such as mosquito, ticks, etc. Here, Japanese encephalitis is known as vector-borne disease because it is transmitted by the vector mosquito. Whereas a zoonotic disease is one that is transmitted between vertebrate animals to humans. Here, pigs and birds are the vertebrate animal. From this vertebrate animal, this disease is transmitted to human. The pigs and birds are the natural host and humans are accidental dead-end host. The disease is caused by Japanese encephalitis virus. Here the zoonotic cycle of Japanese encephalitis is shown. Now coming to the part of the host of Japanese encephalitis. The pigs and birds, particularly the birds belonging to the family, are deity, for example, cattle aggregates, pond hinner, etc. are the natural host. Pigs are reservoir of infection and are often called as amplified host in the transmission cycle, whereas water birds are the maintenance host. On the other hand, human and horses are dead-end host. As when these horses and humans are infected by Japanese encephalitis virus, they do not develop high level of blood viremia. As a result, they are unlikely to infect the mosquitoes. Next part is the agent of Japanese encephalitis. Japanese encephalitis is induced by infection with Japanese encephalitis virus, which belongs to the JEV serogroup in the genus Flavivirus and the family is Flaviviridae. This virion is approximately 50 nanometer in size and spherical in shape. The genome is single-stranded positive sense RNA genome of approximately 11 kilobase pair in length. The virion contains 7 non-structural proteins such as NS1, NS2A, NS2B, NS3, NS4A, NS4B and NS5. The structural proteins are nucleocapsid or core protein, non-glycosylated membrane protein M and glycosylate envelope protein E. It presents an enzootic cycle involving mosquito and amplifying vertebrate host. The next part is genotype and geographic spread of the Japanese encephalitis virus. After complete nucleotide sequencing and molecular characterization of JEV genome, 5 different genotypes have been identified. These are from G1 to G5. All these 5 genotypes have originated from common ancestor from Southeast Asian countries, that is Indonesia and Malaysia. Genotypes 1B and 3 are commonly seen in temperate Asia. Genotype 3 has also been reported from various tropical areas, whereas genotypes 1A, 2, 4 are seen in tropical Asia. The next part is the vector of Japanese encephalitis. Japanese encephalitis virus has been isolated from over 30 species of mosquito, such as the genera Aedes, Anopheles, Culex, Mansonia, etc. The Culex tritonurhynchus is the primary vector in most endemic areas and is associated with rice production, where wetland rice fields and similar irrigation system provide an optimal habitat for larval development. The rice fields provides greater prevalence of this species than natural breeding sites. The Culex vishnui subgroup and the Culex tritonurhynchus, Culex pseudovishnui and Anopheles subpictus were the main mosquito vector and secondary vectors in India. Now coming to the signs and symptoms. Most Japanese encephalitis virus infection are mild. That is fever, headache. Sometimes it may be asymptomatic. But approximately 1 in 250 infections result in severe clinical illness. The incubation period is between 4 to 14 days. In children, gastrointestinal pain and vomiting may be the dominant initial symptom. The severe disease is characterized by rapid onset of high fever, headache, neck stiffness, disorientation, coma, seizure, spastic paralysis, and ultimately death. The case fatality rate can be as high as 30% among those with disease symptom. Of those who survive, 20 to 30% suffered permanent intellectual, behavioral, or neurological sequelae, such as paralysis, recurrent seizure, or the inability to speak. Note that in case of Japanese encephalitis, rash is absent. Now the epidemiology plot of Japanese encephalitis. The first case of a Japanese encephalitis viral disease was documented in 1871 in Japan. Over 3 billion individuals live in JE epidemic and or endemic countries. JV is the main cause of viral encephalitis in many countries of Asia, with an estimated 68,000 clinical cases every year. Although symptomatic Japanese encephalitis is rare, the case fatality rate among those with encephalitis can be as high as 20% to 30%, with neurologic and psychiatric sequelae observed in 30 to 50% of survivors. Japanese encephalitis primarily affects children because of lack of cumulative immunity due to natural infection. Most adults in endemic countries have natural immunity after childhood infection, but individuals of any age may be affected. Next part is the epidemiology of Japanese encephalitis in India. In India, Japanese encephalitis was first recognized via serological survey in 1950s. The first JE outbreak was reported in West Bengal in 1973, followed by reports in southern, eastern and western states. The estimated JE incidence rate was 15 per 1 lakh in 5 to 9 year olds in Tamil Nadu, which is a state in southern India. The laboratory test confirmed that JE cases occurred throughout the year, with more cases in the rainy season. The Culex vishnui subgroup, the Culex tritiorhynchus, Culex pseudovishnui, Anopheles subpictus are the main mosquito vector and secondary vectors in India. Now coming to the part of prevention and control of Japanese encephalitis. Protection from mosquito bite by use of mosquito repellents, mosquito net, and to wear shorts of long sleeves and trousers. Next part is the pig sites. Cattle shells should be at least 3 km away from the human habitation. This 3 km range is the beyond the flight range of mosquito. The community awareness such as media, radio, TV, miking, IC. Breaking the channel of transmission. Anti-larval activities by source reduction of de-winding ponds, removal of submerged grasses using herbicide, which are the ideal site for larval development. Another thing is that larvicide spray that kill the larva, such as abbot and temifos. Biological control of larvae by Gambusia guppy fish. Also use of biocide. Anti-adult measures. Fogging with insecticide. Another important thing is strengthening of the surveillance system to know the occurrence about the disease burden and to control the disease. Next first is vaccination. vaccination of human is the most effective means of preventing Japanese encephalitis. Treatment. There is no antiviral treatment for patients with Japanese encephalitis. Treatment is supportive to relieve the symptoms and stabilize the patients. Nutrition and good nursing care for unconscious patient improve the prognosis. Diagnosis. The Japanese encephalitis virus specific IgM antibody in a single sample of cerebrospinal fluid or serum using IgM capture ELISA. Testing of cerebrospinal fluid sample is preferred to reduce false positivity rates from previous infection or vaccination. Now, coming to the conclusion part. Although JE remains a prominent public health problem in Asia, the reported cases are decreasing due to JE immunization, especially in India and in the People's Republic of China. Another thing is that agricultural pattern changes, urbanization, and improved living conditions. As JEV are likely remain in nature and the risks of JE will persist, it is important to implement the strategies that strengthen JE surveillance system in order to understand the disease burden and apply vaccination protocol in national childhood immunization programs successfully. Thank you. Please like, share and subscribe to this channel.